Good morning, and thank you for being here. I'm Philippe Chambon, the CEO of EG427, and we're about delivering smart DNA medicine at the right place. I'm skipping this. So what we aim to do is to treat chronic disease that have a local root in a safe, repeatable, durable manner at the point of need. And how we do it based on an herpes platform. We're using non-replicative herpes vectors to enable us to deliver complex DNA sequences, as you will hear. Our first product, EG110A, silences the function of a subset of a neuron, type C neuron. We have a second product behind this who does something very different. That one changes the function of excitatory neuron into inhibitory neurons. The company was set up in late 2019 out of technology from a European consortium of university led by University of Versailles, as well as Hôpital de Garches, which is the major trauma center in France for spinal cord injury patients. We are based in Paris, 15 FTE at this stage, mostly in the research lab, and we've raised a bit more than 15 million euros, mostly in terms of Series A money. I have a great team working with me. Samuel Salo is our Chief Development Officer. He's responsible for the activities in preclinical. Alberto Epstein is one of the scientific founders of the company. He is our Chief Scientific Officer and is one of the leading lights in the biology of herpes viruses. Cornelia Haig-Molken-Taylor is our Chief Medical Officer. She has a truly unique background for what we're trying to do because she is the person who developed the urology application of Botox at Allergan, and then she was the Chief Medical Officer at Eurovant, where she developed a series of their drugs, which led to their acquisition by Sumitomo. Our VP of Research, Greg Sarek, is the person that has put together what I think is a very innovative vectorology platform, and Michael Contrastin, our VP of Strategy, is leading the effort to build our IP portfolio as well as partnering. We are seconded around for all of this by a great team of advisors that represent some of the leading expertise in the development of herpes vectors, be them oncolytic or non-replicative, as well as the biology of herpes, in particular host interaction. So why herpes vectors? Herpes vectors are, in our views, very powerful tools to overcome the current limitation of gene therapy. And that's because they are very large viruses in which you can put large transgene, over 30 kb of material, as well as complex promoter sequences. They stay as episome in their host, in the nucleus of their host cell, where they are expressed basically for the life of the cell. So we can expect to have very long efficacy in patients. They also show low immunogenicity, which means we don't need to test for antibody, and we can also do repeat dosing with those type of vectors. They are easier to manufacture because it's an infection process, not a transfection process. And we have very experienced regulatory body by now, given the activities in both oncolytic herpes virus and non-replicative viruses, in particular led by Crystal Bio, which should have its first product approved by the FDA next month. So with those type of tool, in our case, we're first applying them to the peripheral nervous system, and in particular, disease or dysfunction of the bladder. But we're also looking at a series of the other pathology driven by the peripheral nervous system. And what's notable there is, in fact, the therapeutic options are often quite limited or non-existent. So we should have worked for a long period of time. Our lead project, EG110A, is a vector that expresses the light chain of botulinum toxin, and it does that under the control of a promoter-specific for type C neurons. So we get very selective expression only in a subset of those neurons that leads to the inhibition of the activity of those neurons. Type C neurons are one that you all well know, in fact, because when you eat hot chili pepper, they are the neuron that reminds you that it burns. So they are neurons that are responsible for sensation of heat, burn, itching, cramps, and the like, and they are present across a whole series of organs in the body, in particular the bladder, where they are responsible for the initiation of contraction and micturition, which is what we're exploiting here by inhibiting their activities. I'm going to try to get the video. So now to show you what neurogenic detrusor overactivity is, I'm going to try to make the video work. And yes, it is. So that's the normal bladder. You see it filling, and you see the activity of the sensory neuron that transmit their signal to the central nervous system, comes back down, activate the motor neuron, and you have voiding of the bladder. That's a normal person. Person with a spinal cord injury, you just have the sensory neuron shooting up, and it comes back down to the motor neuron and generate in uncontrolled contraction and uncontrolled voiding. For us, we'll be injecting our product in the bladder wall, as you see here. It will then migrate to the neuronal body in the dorsal root ganglia, where it blocks the activity of those neurons and hence restore a more normal micturition process. So that can apply across a whole series of indications. Our initial indication is neurogenic bladder, but you can also think about overactive bladder and the like, and this results then in a very large market opportunity. And we're doing that in the context of a very well understood regulatory pathway because that is precedented by the development of the botulinum toxin for that indication. And what we know is we need to show efficacy across two indications, spinal cord injury as well as multiple sclerosis, in order to have a broad labeling that will apply to patients irrespective of whether it's a spinal cord injury or a Parkinson patient or a stroke patient and the like that have that type of disease. So that brings us also back to a very large opportunity. What you see here is what happens, what we've shown in our animal model. We inject our vector in the bladder of a rat in the bladder wall. That's the brown dot that you see on the left side. From there, it migrates through the neuronal axon to the dorsal root ganglia, and there it stays as an episome in the right cells. And we know it's the right cell because we've done a whole series of histopath experiments, and what I'm showing here is just one cut of a ganglia where we are visualizing a type C neuron that expresses within it the R transgene BONTEF. And so we have proven here that we're able to engage our target and which should result in inhibition of their activity given the presence of botulinum toxin F inside those cells, but only inside those cells. But more importantly, we have animal data that validates what I just said. We've been running a whole series of animal models. This is one of the latest one, which is an overactive bladder rat model. The way it's run is we inject a treated group of animal with our vector. We have a control group. We wait 5 weeks, and then we perfuse the bladder of those rats, first as control, and second by adding capsaicin in the perfusion, and that, of course, irritates the bladder in a major way. And we look at the change of contraction in those bladder. And what you can see here very nicely is that we increase the interval between contraction in the treated animal versus the untreated one, or to put it in a different way, bring back the bladder more toward normal in terms of contraction behavior. And we were very happy with the result because what we show here is a very strong p-value of p=0.001 from an efficacy perspective. As importantly was the safety that we saw in those rats. We had no change in body weight over this 5 weeks period in our treated animal versus control. And more important for us from a mechanism of action perspective, we didn't see any change in the bladder avoiding ability versus the control. And so we had no residual volume of urine in those bladder, which is important because that's one of the differentiating aspects versus Botox type of treatment, where because the botulinum toxin act on the neuromuscular command, they do have that as a problem. So what are the next steps for EG-110A? We have had already positive pre-IND meeting with the FDA as well with MHRA in the UK. We are finishing the CMC manufacturing of our clinical lot, and we are processing our toxicology study, all of this in order to put together an IND that will be submitting in the first quarter of 2024. In parallel with developing our clinical development plan, we have our first in-man protocol, which will be a phase 1b/2a study already designed. We're selecting the CRO to do the work for that, and we're also expanding the clinical development plan to cover multiple indications besides neurogenic bladder. The next one after that is going to be overactive bladder, in particular given the result that I just showed in animal model. Behind this, we have a second program that really shows the versatility of this type of vectorology effort in terms of modulation of neuronal activity. Here, what we do is transform excitatory sensory neuron that express glutamine into inhibitory sensory neuron that express GABA. And we do that by having a vector that which transgene is GAD67, which transforms glutamine into GABA. And that way, we're able to move from the excitatory function to the inhibitory one. And the application of this is, in fact, spasticity. There's a precedent for the mechanism of action. It's a drug called baclofen, who also increase GABA level, but baclofen has a lot of side effects and complexity linked to it. It's used in spasticity. The other major therapeutic alternative in spasticity is, again, Botox, but itself has its own series of challenges in that indication. But that being said, it remains a very large opportunity there from a market size perspective. So we are at the stage where we're doing animal study to validate that, in fact, we can get very targeted efficacy on a local basis. With all of this, we're in the process of building a very strong pipeline. Besides our two products, we're also investigating pain models to identify a vector in particular for neuropathic pain type of settings. We're also looking at the inner ear as a potential target for herpes vector, where there's a place where there's a big need for innovation, and we believe that if we can get the herpes vector to the hair cell, we may have the opportunity for a broad range of development activity. And lastly, we have a project where we change the tropism of the vector by changing some of the glycoprotein on its surface, which then will enable us to target different type of tissues. So with all of this, we're in the process of building a strong IP position. Our initial group of patent licensed from the university have started to issue in Europe, Japan, Israel, and Australia, still pending in the US. We have filed other patent, in particular on our next generation vector backbone, and we are also working on continuous cell line improvement, process improvement, and so on, because one of our goals is to get, obviously, to the lowest cost of good possible, given that here our aim with this platform is not to target rare disease, is to target chronic disease with reasonably large patient population. So our price point has to be compatible with that, and hence cost of goods is a major driver of our activity. Fortunately, the type of infection process that we're utilizing seem to enable us to get to very low cost of goods at scale. So in summary, we believe that herpes vectors are a great next-generation tool for genetic medicine. We have shown through 2 different vectors amazing ability to change the function of different subtype of neuron. Our first project will be in human clinical trial in 2024. We've built a whole vectorology team that can be the basis for partnership effort. Thank you very much for your attention.